is an Dr. Revzen is an associate professor at with appointment in the electrical engineering and computer science department and also in EEB. His work has focused on bio-inspired robotics and the comparative and functional biomechanics of legged locomotion. He has done extensive experimental work on both animals and robots, as well as work in applied mathematics in the field of dynamical systems. And today he will be talk about how walking is a lot like slithering. So after the talk, there will be a Q&A section. So if you have any question, please type that in the chat box and I can help read them. Or if you want to ask the question by yourself, you're also welcome to unmute yourself and just speak up after the talk. And now let's welcome Dr. Revzen to give us his talk.
Thank you. It's a pleasure to give a talk to EB. And I should mention that this work is in collaboration with folks from other universities, Glenna Clifton, who recently moved universities, and Nick Gravish, and my students, Brian Bittner and Dan Zhao. I write in collaboration rather than co-authors because I am to blame for everything wrong with this talk, but the good ideas are from them. I run the Bio-Inspired Robotics and Dynamical Systems Lab, or BIRDS Lab in EECS. It has nothing to do with birds because I work on legged locomotion. But just to give you a sense, we study a whole bunch of animals. These are some of the animals I've worked on over the years. And we also build a lot of robots. You see some of them over there, and I won't talk about all of those. And then in the middle, there are a whole bunch of mathematical methods that we've developed to elucidate principles from the animals and then carry them over to the robots. And I will shamelessly plug that occasionally I also use my courtesy appointment in EEB to give a seminar, and that's an EB 800 seminar in comparative biomechanics. And if you're interested in the biological end of what I'm going to be talking about today, please come to my seminar when I next teach it, because that's the sort of thing we do. So I'll just summarize my whole talk in one slide, and then anyone who is not interested can leave. The talk in one slide is saying this: these bugs, these animals with multiple legs, are functionally equivalent to slithering animals. So in a more specific way, in the sense of being governed by a mathematical object called a connection, multi-legged animals and multi-legged robots are like slithering snakes and swimming nematodes. An important note, that doesn't apply to us humans. It takes having at least 3 legs on the ground at the same time. So unless we're crawling, this doesn't apply to us, which might be why it hasn't been discovered sooner, because it's just very unintuitive to us. So here's a broad outline of my talk, and some gratuitous ant videos to liven it up. I'll talk a little bit about slipping and dynamic locomotion, and how slipping doesn't necessarily imply dynamic motion. And then about various systems being Stokesian. And what I mean by Stokesian is following the same kind of math as low Reynolds number swimming, which is sometimes called swimming in a Stokes fluid. So let's start. This I think is most people's notion of slipping. I'm very impressed by Sir Chaplin's ability to perfectly execute a judo breakfall. It's a pretty dynamic maneuver. But if we add a few legs, we might get this guy. And he seems to be doing a lot better with his slipping. And certainly things are a lot less catastrophic for a quadruped. And if we go to a hexapod of the sort I worked on, which are cockroaches, you see that even though it is slipping a lot, as soon as the momentum is sort of canceled out, the cockroach is managing to sort of swim along the surface with a great deal of slipping. So how much slipping is actually happening with animals? Well, here are 2 animals and 2 robots that have multiple legs on the ground. And the thin traces correspond to the motions of the tips of the feet, the tips of the tarsi on the cockroaches and the feet on the robot through space. And the thicker areas are when those feet are on the ground. And you see that for the ant, the feet don't move much when they're on the ground. For the cockroach, when it's running, they move quite a bit more, about comparable to the robot on the top right. And then the even more multi-legged robot on the bottom right has even more slipping. So part of what I'm going to try and convince you today is that when friction dominates locomotion, which is what happens in all of these cases, you get a kind of non-dynamic motion. And there are actually three classes of this non-dynamic motion that we know about. So the most uh, uh, simple to understand and basic is what's called a piecewise holonomic or non-slip motion. I'll explain that a bit better later, but it basically corresponds to legs remaining in place on the ground and the body changing shape. Then we have what are called Stokesian or principally kinematic motions in the geometric mechanics literature, and those correspond to things a lot of biologists learn about in their undergraduate classes, like bacteria swimming with bacterial flagella, or small animals swimming in very what is effectively a very low Reynolds number fluid. And there's a third category that is very similar to the Stokesian motion, which one of my students proposed and basically we discovered, which is what is called perturbed Stokesian motion. I'll mention that as well. So why should we as biologists even care about this? Why should we care about motion being Stokesian? Well, Stokesian motion has a very special property, and that is that the motion of the body through the world depends only on the shape of the body, on the sequence of shapes, and not on the speed in which that sequence of shapes is executed. So if you're Stokesian, doing a motion slowly or doing it really quickly will give you exactly the same outcome, just with a different time scale. Uh, so that implies that in terms of the nervous system, only tonic or position sensing is needed to control that motion because the speed doesn't matter. It also means that if we want to know how we navigate through the world, we don't need exteroception. We don't actually need to see any markers in the world to know where we are, because we only move through our own shape change. And if we can register our own shape change, we know how far we moved. And so that could have pretty profound implications, both for the evolution of motor control and locomotion, and to colonizing environments where exteroception is challenging, for example, in the dark or in murky waters, um, or other situations where you can't actually see landmarks to know how far you've moved. Uh, so let's dig in a little into understanding locomotion better. So this is basically true of all moving animals, that they cycle the body shape, and that leads to motions in the world. So if you look at this cockroach walking across the screen, and imagine that you're just tracking the animal with your eyes, you're moving with the animal. The shape of the animal is changing in a cyclic fashion. It's going through a repeated pattern of motions. But that repeated pattern of motions doesn't cause a repeated set of locations for the animal. The animal is actually achieving a net motion to the side. And so from a mathematical standpoint, people sometimes would call this a piecewise holonomic motion. So what's holonomic about it is that if I wanted to know what the body frame is over time, so the frame of the center of mass as a function of time, it's actually describable as a body frame which is a function of the shape of the body. That is, if I know the shape of the body, I know how far the body moved with respect to the feet that are planted on the ground. And that sort of relationship, where there's a relationship between the shape and the center of mass position, would be um, a kinematic relationship. But that sort of relationship can also be rewritten by just applying standard rules of calculus and taking the derivative of both sides as the equation on the right. and that is that the body velocity is related to some matrix, in this case df, the derivative of that function f, multiplied by r dot, which is the rate of shape change of the body. So what we're really seeing here is that in this piecewise holonomic case, we can write things in terms of body velocity is some linear map, some linear relationship to shape velocity. If we go faster in shape, we're going to go proportionately faster with the body, but in the same direction as we would have gone before. And then what is it that enables us to move? Well, obviously, if that was the whole story and R repeats itself, the shape of the body repeats itself, then the position of the center of mass would repeat itself and we wouldn't move. So what enables us to move is that the support itself switches. There are times where the cockroach switches from 3 legs to another set of 3 legs, and again 3 legs and another set of 3 legs. And those switches cause a discontinuity where we're really thinking of the shape of the body as switching, and switching in the sense that the set of support legs has switched from some time t minus instantaneously to some time t plus, and these jumps are what enable us to move. But it turns out that there's another way in which you can get a very similar relationship, and that also has been known for a long time and in part developed by folks who are here at University of Michigan, like Tony Block. And that is what is called Stokesian motion and has been used to study slithering. So this is a nematode swimming along. And of course, the nematode is swimming smoothly. There aren't any resets like changes in support legs for the nematode. But these velocity relationships still exist. So unlike the case we had before, where the velocity depended on some derivative of a function, this df, there is some other matrix that depends only on shape that takes our shape velocity into our body velocity. And that matrix is called a connection in some of the literature. And this sort of motion is called Stokesian motion because the examples people usually use for it are examples of moving in a 0 Reynolds number fluid, a very, very viscous fluid, or sometimes what's called a Stokes fluid, and hence this is called a Stokesian motion. There's some of the literature which will also refer to this as geometric motion because of the fact that it is only the shape of the motion that governs how far you move. It's not the rate. So to compare that to something that's very dynamic, imagine a frog jumping. If the frog has a higher takeoff velocity, it's going to fly further. Just knowing the shape of the frog doesn't tell you how far that frog will move. But for all these Stokesian movers, if they go through the same sequence of shapes, they will end up moving in the same way through the world. So let's see in a bit more detail how nematodes are Stokesian. And for that, we need a model. So a classical way to do that, and what we did as a prelude to our further work, is we got a nice data set of swimming nematodes from some friendly people at University of Pennsylvania. And then we used what's called a Purcell swimmer model, which is a particular kind of Stokes fluid swimming model, and calibrated the parameters of that model to make it correspond as closely as possible to the observed motions we had from our nematode. And once we have that model, we can use it to discuss what would happen if the worm was swimming in some other form, in some counterfactual shape change, doing something that worms don't actually do. So to convince you we have a good model, here's the data plotted. So we have the X and Y of the center of mass plotted in green for experimental data and blue for our model, and theta the angle of the body. And notice that theta corresponds a bit less than X and Y, but you still end up with something pretty good. And let me show you that in a more intuitive form as this video. So again, green is our observed worm, blue is our simulated worm. And you see that they move very similarly through space. So now we have a good model of the physics of a swimming nematode, calibrated to the nematodes we have in our data set. We can study some interesting locomotion questions, like what is it that C. elegans actually optimize when they swim? Do they, for example, optimize swimming as far as possible in each cycle? Do they try and save energy and swim as efficiently as possible, or what other things might they optimize? So we have the first bullet. We've constructed an accurate physics model. And now we can pick a goal and compute the motions under our physics model, compute the dependence on the parameters of the motion, improve the motion, and repeat. So basically do an optimization loop to try and optimize for our goal and see whether the motion we end up with looks a lot like the natural motion or not. Except there's a problem. When you describe the motions of a soft body, you're going to have hundreds of parameters, but these simulations are pretty slow. And sometimes, which we also care about, we don't have a good physics model, a good general physics model. So instead, we wanted to improve this approach in the following form. We replace our accurate physics model with a fast to simulate data-driven model of the physics, which is going to be valid only for the given behavior. So now our loop looks a bit different. We have some motion, we generate a fast model for it, and then look at the dependence of that fast model on small changes to the parameter, improve the motions, obtain a new model for moving around this new behavior, and go through the loop in this way. And now I'm going to get to the really only equation heavy slide that matters at all in this talk. And that is, how do we do that? Well, take our connection model, AFR R dot. R is the shape, R dot is the rate of shape change. We split the parameters into theta, which is where we are on the characteristic cycle that the animal is exhibiting, and delta, which is a perturbation, a small change from the typical motion. Now, because we split R into theta and delta, we also need to split R dot into theta dot and delta dot. And now we multiply things out, and we take this nonlinear function A, and we just do a first order approximation for it, collect the terms, and we end up with a linear regression, a sort of GLM model of the sort that anyone who's taken a biostatistics class has encountered. So for every point on the cycle, every radial line you see over there, we have one of these generalized linear model regressions. And this gives us a model of A, which is valid only in the neighborhood of the characteristic motion that we've seen. But turns out that's a really powerful thing. And to give you some sense, we could use this to optimize this swimming model, where we start with a fairly ridiculous way of this 9-segment swimmer wiggling around. And we let it build a local model, do some improvements on that model, build a new local model, rinse and repeat. And within about 10 cycles of this process, which basically corresponds to what would be 10 experiments with a robot, we can optimize and get this really nice serpentine motion, even though we're optimizing over a space that has almost 300 dimensions. So this is an extremely rapid way to optimize a gate. And it turns out that under varying conditions of noise, from really low noise to very high noise, we always converge to the same cost of transport, which is this same slope here. Only when we have larger noise, we converge to solutions that have larger motions. And when we have smaller noise, we converge to solutions that have smaller motions, but they're all equally efficient for an optimization that's trying to optimize cost of transport. So we have a tool now to examine what nematodes would do if they were optimizing something different from what they've done in nature. And here's what we actually get. So if nematodes were trying to optimize their displacement, move as far as possible in each cycle, they'd be doing the blue motion. If nematodes were trying to save energy, they'd be doing the magenta motion. But what they actually do is the green motion, which is a bit more like the magenta motion than the blue motion, but not conclusively either. So what you can draw from this is that we have this tool that allows us to create a data-driven Stokesian model and then use that model to query behaviors and optimize them. Now, I should say we had this complicated regression, but that regression that we did was based upon the assumption that we're moving with a connection in a basically zero Reynolds number or Stokesian fluid, which doesn't actually appear in nature. That's just a limit of relatively high viscosity. So one of my students pointed out to us that there's actually a way to extend this approach to small Reynolds numbers that are still non-zero. And I'll just point out that it boils down to adding a bunch of terms to the regression. There are 3 more terms that need to be added to the regression, to that GLM. And then you should be valid for small but non-zero Reynolds numbers. And to show you how well that works, here are 3 simulations. Red is a fluid dynamics simulation. Blue is what you get if you assume it was a Stokes fluid. And green is what you get when you add the additional terms. And the graph you see on the left is looking at the log of the Reynolds numbers. There's a range where all these lines bump up, which is telling us our added terms fit the data better than at very low Reynolds numbers. They basically perform the same way as a Stokesian model. At very high Reynolds number, neither model captures things very well, so we're doing no better than a Stokesian model. But in the intermediate, the perturbed Stokes regime actually gives us a better model for how a swimming system would move. Okay, so I've shown you some tools for how to model this Stokesian swimming of a nematode, and I promised to talk about things with legs. So let me try and convince you that ants are Stokesian as well. So the question we're facing is really this. We know that non-slip walking is piecewise holonomic. We have all these systems that we know are Stokesian. And in fact, for nematodes, we've built a model and shown that it matches things pretty well. Where do ants sit? They may well be in here, in the piecewise holonomic regime. We have a tool for building Stokesian models, so that catches a slightly broader domain. Let's see if that tool enables us to understand ant locomotion well. And here step in my amazing colleagues, Glenna and Nick, and their ant data processing pipeline. So they have these arenas in which the ants are running, and they can collect thousands of trials and then use a computer vision system to auto track the tips of the legs and the body of the ant. We then take their time series. We use an algorithm I developed a few years ago to assign phase to figure out where we are in the motion cycle of the animals. That allows us to obtain a limit cycle. What is the periodic behavior that the animals are executing? And also using that information to do some imputation, because there are always gaps and missing measurements. We're, of course, pretty careful with that. But eventually we get this huge corpus of ant locomotion data, which we can then partition and analyze. So here's what the auto tracking looks like. And I think this is super cool, maybe because I spent so many years clicking on animal videos and suffering horribly, and now no one needs to do that anymore. So what we get is the ability to collect body, antenna, and foot motions. They also have data on multiple terrains. I'm not going to talk about that here, but I will point out that our data set corresponds to about 2 million frames. So definitely a very large data set. And the first thing you want to do when you have a data set like that is do some sanity checks. So what we do is we partition the locomotion data based on speed into 4 quartiles. We're ignoring the slowest speed because ants do a bunch of different behaviors when they aren't moving, and look at the top 3 quartiles. And then we split the data also by whether the ant is turning right, left, or walking straight. And this isn't just in terms of 33%, 33%, 33%. And one of the first things we'd expect is that ants being bilaterally symmetric, the motions they execute when they're walking to the left are mirror images of the motions they execute when they're walking to the right. And sure enough, this is, of course, not the entire information, but this is just motions fore aft along the body. You see that that this is indeed true. That is the means and standard deviation bands of the motion match pretty well walking right and left. Okay, so we have one sanity check. Now let's ask ourselves something that would have to be true if the system is actually Stokesian, and that is that the animals would go through the same shape changes at all speeds. They move through the same geometry, but they do it in different rates. And sure enough, when we take these 2 categories, the foot motions look pretty much indistinguishable. That is, if I showed you the sequence of frames and didn't show you the timestamps, you wouldn't be able to tell me if the ant is walking faster than average or slower than average. But if I actually looked at the velocities, the differences are pretty clear. That is, the velocity as a function of phase is very different in the slow region in blue versus the fast region in red. And remember, these are velocities in the body frame. That is, this is how fast is the ant changing its own shape. Okay, so we have some sanity checks. Now let's try and use our fancy tool that builds these Stokesian models to predict some of the behavior. So the next 2 slides are probably the most important ant result slides. And both of those slides are about a model that was built from that one little, my finger's disappearing, from that one central square between the red and blue. That is, ants walking at middle range speeds and not turning right or left. So we take that collection of data, the ants doing their average behavior, and then we build a connection model for those data. And using that connection model, we ask ourselves, is this actually capturing enough information about how the ants move? That is, if we only saw the ants doing this average behavior, have we learned enough about the ant environment interaction to extrapolate to all the other behaviors we observed? So let's see what we get. So here we compared 3 models. The model in light gray is just our periodic behavior. That is, as a function of phase, we build a Fourier series model which says this is the typical shape of the ant as a function of phase. It does a lot better than just assuming that the ant is walking on its mean velocity. So if you want to know how fast the ant is moving, it predicts the speed, but it doesn't predict the speed too wonderfully well. For ants walking at average speed, it predicts about 14% of the variance. For ants walking faster or slower, it does pretty poorly at predicting speed. Now, that's sort of unfair because obviously if we're taking a model that's moving at average speed, we should expect that it will be at a different speed than the fast or slow. So the middle model is taking the average rate of shape change and using it to rescale the speeds that we predict. So all we're saying is, well, this ant is moving faster on the average. So let's assume that all the speeds are faster by average. And sure enough, that works pretty well. That improves the prediction quite a bit, especially when we're changing speeds. And it turns out that our Stokesian model does about as well as that. So when it comes to predicting how fast the ant is moving forward and back, the Stokesian model is about as good as just saying what's the average speed of the ant and extrapolating that if the ant is changing its shape fast, then it should be moving forward fast. But here's where things get really interesting. If we want to predict something that we haven't seen, remember this middle square here has no turning in it. We can get nothing out of these two basic models to explain the turning behavior, but we're getting a prediction of over 60% of the variability of, or on the order of 60% of the variability of the turning rate. That is, even though we saw data that didn't have any turning, we understand the interaction of the ant with its environment so well that we can predict turning pretty well. So this is telling us that we have a pretty good model of ant motion using our Stokesian approach. Now, granted, that Stokesian approach would also apply to piecewise holonomic motion, so it doesn't differentiate between those two possibilities. So what we've shown here is basically this. Ants are either piecewise holonomic or Stokesian. Now, anyone who took a little bit of undergraduate biomechanics or knows a little bit about low Reynolds number swimming knows that that usually corresponds to things being small. Unfortunately, we don't have really large insects. So this is where we switch to robots and ask ourselves, does this idea about multi-legged motion generalize to larger multi-legged systems? So the question we'll address next is, are multi-legged robots that have 6 or more legs Stokesian as well? And so for that, let me introduce one of our robots, and you'll excuse me for having bits of this video that have me and my students in it, but you see the robot in the background. Especially I want you to watch the next segment coming up now, where you see the student falling on the snow where the robot is going pretty well. So these hexapedal robots do spectacularly well on rough terrain. In fact, better than humans in some ways, even though they're exceptionally simple machines. And of course, the first sanity check that we're going to check for these robots is, do they follow the same shape at different speeds? The exact same sanity check we did for the ants. And sure enough, at different speeds, which are at different colors here, the robot is executing exactly the same shape change with respect to its body. There's a tiny difference on the far end for the front legs, because they hit the ground and bounce a little when the robot is moving faster. So now let's see if that holds even when we're executing more complicated behaviors. So here we're setting the robot to turn with a turning parameter that changes the shape of the motion of the legs. So there are 3 values of the turning parameter, but then we execute each of these behaviors at multiple speeds, and you see that the behavior ends up grouped by turning parameter. So we have the 3 turning parameters, but at each turning parameter, we have both yellow and blue colors, or warm and cold colors, which is telling us that the speed with which we executed the motion doesn't actually change the outcome. So this is sort of the direct opposite of a very dynamic motion like a frog jump, where you get a completely different outcome if you jump quickly versus you jump slowly. If your takeoff velocity is high, you go very far. If your takeoff velocity is low, you go a short distance. For a Stokesian motion, the speed doesn't matter, only the shape. Another way to say that is that if we look at how far we travel, or how far we turn, say angle per cycle, it shouldn't depend on speed. And so we can do that as a regression problem. And here you see data looking at the 3 different steering parameters we had, the range of rotation rates that we observed, and regression lines through them. And the point I want to make is, even though the box plots on the left tell us that sometimes these slopes are significantly different from zero, those are bootstrap significance tests, these slopes are very low. These lines look pretty horizontal. They're not intersecting zero, which is what you would get if you were hopping. If you were hopping, your velocity is zero, you don't travel at all. Your velocity is high, you travel far. This is very different from what we're getting here. We're getting almost constant result at various speeds. So that's for one hexapedal robot. Let's try and be a bit more ambitious and look at a great variety of different numbers of legs. So this is our multipod family of robots. You see them going all the way up from 4 legs to 12 legs. And what we do here is we explore both the number of legs and the impact of changing the phase difference between consecutive legs. That is, how far apart in time are consecutive pairs of legs. So if the difference is small, it will look like a long undulation, and if the difference here, let me start that again. And if the difference is large, then you'll get something like what we see in the middle 2 videos here and here, where consecutive pairs of legs are almost in antiphase. So what do these results look like? Well, it turns out they fall on a single master curve. So remember, now here we have a lot of slipping, right? These legs slide on the ground a great deal. They're definitely not piecewise holonomic. But all these robots at different sizes fall on the same curve, where as a function of the phase difference between consecutive legs, this is the x-axis, the distance traveled per cycle is almost the same regardless of the number of legs and regardless of the frequency. So the plots you see on the right show you the offset from this mean behavior, the blue line in the middle, as a function of the frequency with which the robot is moving and as a function of the number of legs. And you basically see that for all numbers of legs and all frequencies except the highest, we don't really see a big change. So this entire family of robots is moving geometrically, is moving in a Stokesian way, where it is only the shape change that governs how far we travel, even though we're slipping a lot. So that's pretty strong evidence that these robots are, for all intents and purposes, slithering, even though they're walking on legs. And in case you're wondering where are my cockroaches who have worked on my entire career, well, the cockroaches also show up. So here's what that data is like for cockroaches. Again, the slope is pretty small. Interestingly enough, it's negative. So what we see with the cockroaches is that the distance they travel per cycle of their legs actually slightly decreases the faster they're moving. So the faster they're changing the shape of their body, the less they move per each cycle of motion, which is kind of surprising. But again, that slope is pretty small. It's a lot closer to being horizontal than it is to be anything like hopping. Okay, so at this point, it's rather tempting, at least to many engineers, to try and build a mechanistic model of what we're seeing. And so we'd like to see how the forces on the legs generate this motion, and the natural choice there would be to build a model with Coulomb friction. And really what I'm trying to point out here is it's a model like this. We have springs playing the roles of legs. These springs are touching the ground and we're sort of moving them around, and they're sliding on the ground, generating the friction forces you see right here. So the XY forces for each leg are just coefficient of friction, normal force going in the opposite direction of velocity, basically your high school level of friction. Turns out this doesn't work at all. That is, this will not generate something that looks like a connection. All the pieces here don't fit. And so we're kind of stuck until we came up with an idea, and physicists sometimes call this an ansatz, where you basically postulate that the math doesn't match the physics you thought. You just give it the form that will make things work out. And so what we did here is we changed the force to also be proportional to the magnitude of velocity and not just directed in the opposite direction from velocity. And then all these equations take the right form. We get a connection-like term. And when we plug this model into our robot data, we get pretty surprising results. So what you see here are theta dot y dot x dot data for another version of a robot, a bit like the multipod. We see a great match between the model prediction and the observed data. We see a great match for the big ant robot. In fact, that was so surprising to us because the friction model doesn't match what we'd usually expect in physics. We asked our colleagues to give us data for another robot, for their robot, which is a quadrupedal robot, and there it shouldn't work at all because we need more than 2 contacts on the ground at a time. It works pretty well for the quadrupedal robot, except for these little spikes that presumably happen when only 2 legs are on the ground. We're not sure. We haven't determined that yet. So we got this pretty good model for robots where we assume that the friction is going to be viscous friction like we get in swimming, rather than Coulomb friction like we're taught in our physics classes. So that's pretty surprising. So let's bring this back to the broader question of how do we classify then motion of animals? What do we learn about the broader animal world and comparative biomechanics using this? So really what I'm saying is that there are different kinds of movement. Holonomic movement, where there's a relationship between shape and location of center of mass, is not really movement, because that's what happens when I'm planted in the ground. Here I'm leaving my feet on the ground and I'm just shifting my body. If I go through a loop in shape, but I don't lift my feet, I won't move at all. But as soon as I can be piecewise holonomic, I can have these discontinuities between footfalls, I can move. I can also have Stokesian motion, which has this linear form. And as it turns out, I can have the perturbed Stokesian motion. The difference here is that there's an additional term, and sort of pictographically here, you see that these lines are straight. There's a linear behavior. In the perturbed Stokes behavior, it turns out that you have something that's not entirely linear. And then you can have a behavior that is completely dynamic, and that's that last figure on the right, where the relationship between shape and body velocity also depends on additional information. And that additional information is the momentum of the center of mass. So when friction dominates the motion, when momentum decays quickly, which is what happens in viscous fluids and low Reynolds numbers, we have the relationships in the middle, one of the 3 relationships in the middle. But if that doesn't happen, then we can have a dynamic relationship. So going from left to right, more and more information is needed to predict the motion through the world. But we can also think about this in the following form. Going from left to right, here we can't move at all. So we basically are always stopped. Here, in these 2 motions, piecewise holonomic and Stokesian, the moment we stop moving, the moment our shape change stops, our velocity goes to 0. So we stop very quickly. Perturbed Stokes, we don't stop instantaneously, but we stop quickly. And then if we're fully dynamic, we might be in the situation of that cockroach getting on the plate where it's just sliding along, and regardless of how it's going to change its shape, it's going to keep on moving. So there's a momentum term, and we can't stop quickly at all. So we're arguing that thinking of how fast you stop in terms of how many cycles it takes you to stop your motion is a good generalization of the Reynolds number into the realm of legged locomotion. So what can happen is the same animal, here a cockroach, can exist in all of these domains. When the cockroach is standing, when the legs are pinned on the ground, it's just holonomic. It's not actually able to move. It walks very slowly. Maybe it's piecewise holonomic, a bit faster. It might be in a Stokesian or perturbed Stokes regime. And then on this vertical axis here, the stopping ratio starts growing. The time it would take the cockroach to stop itself if it freezes its body motion grows and grows. And as that grows, we get more and more dynamic until we're at the most dynamic motion, which is flying, where even if the cockroach suddenly stopped moving at all, it would keep falling. It would keep following a ballistic motion. So basically, it can't stop itself in place at all. So how does this distribute itself over the animal kingdom? Well, we took a whole lot of data from a whole lot of papers and tried to see what governs this stopping ratio, this predictor of whether we are Stokesian or not. And there are two ways we could predict limits on this. The horizontal band you see here is within an order of magnitude the prediction of what the stopping ratio would be if animals are limited by their ability to generate friction with the ground. And the somewhat diagonal band you see here is where animals would be limited if they are limited in their stopping ratio by the muscle force they can generate. And just for giggles, because I am a roboticist as well, we added in the stars a few robots, which, as you see, underperform animals pretty uniformly. And so I think there's an interesting observation to take from here, and that is that it sure looks like for large animals, the ability to generate power from their muscles is governing that behavior. There's a slope that follows pretty closely the allometric scaling of muscle force. And what that is telling us is large animals can't stop because they aren't strong enough. For small animals, it looks like we're limited by friction. That is, small animals can't stop, not because they aren't strong enough to stop themselves quickly, but because they can't generate a strong enough contact force with the environment. And this might give us a hint as to why small animals have really complicated claws and feet and ways of attaching to the substrate. Because if they want to control their motion in this very convenient way that Stokesian physics would provide, that Stokesian motion would provide, they would need to improve their contact forces. Okay, so that was a long ride. Let me get to my conclusions. So what I've convinced you is that multi-legged locomotion is governed by a connection just like slithering. And that is at least for those cases that have enough legs in contact with the ground. And that multi-legged locomotion looks like slithering even when they're slipping. So even though as bipeds, we're sort of assuming that Charlie Chaplin-esque situation where slipping is dynamic, that's not necessarily the case for animals with many legs. They could be slipping a lot while still doing something which is governed by a connection and is not dynamic. Animal locomotion can fall in different classes, and looking at this stopping ratio parameter can help us systematize where they fall. So in the case of swimming, stopping ratio is like the Reynolds number. When it's small, you stop quickly. When it's large, it takes you a while to stop. But it applies also to legged systems. If you can stop quickly, you're probably Stokesian. If it takes you a while to stop, you're probably not. Last but not least, small animals are friction limited and large animals are power limited. And with that, thank you for listening, and I'm happy to take any questions.
Okay, thank you, Dr. Revzen. So now it's question time. I noticed that I don't have access to the chat box. So I don't know if this is true for everyone. But if it is true for everyone, I think you can just unmute yourself and speak up
Yes,
if you have
please.
questions. Yeah.
I promise I won't bite the first person who asks question. The second one maybe, but not the first one.
So Dr. Revzen, can you see anything appearing in the chat box?
I'm not seeing a chat box either.
Okay.
I don't have access to one.
Yeah.
I don't know why that is the case.
Yeah, this is weird. Yeah. Please speak up if you have questions.
I think it might be worth making sure that people can unmute themselves.
Okay. Yeah, they can unmute themselves. Yeah, they have this permission.
Hi, Dr. Revzen. I had a quick question. So this is mostly when it comes to comparisons between models and data. So we saw very close correlations between the data and the models themselves, but there's still like a small gap there. Do you think it's possible to improve the models to close that gap even further? Or is this about as close to the best we can do?
Oh, well, I mean, one would hope that that we're in the realm of Newtonian physics, and if we really got into the details, um, we could fill in those gaps. But I think um, when you talk about animal locomotion, there's so much individual variability, and there's usually so much variability in the environment that it's hard to narrow those gaps very much. I think for the robots in very controlled environments, we might be able to narrow them further. But with the animals, I'm not super hopeful that we can do much more than say predict 70% of the variance instead of 50%. Other questions?
Okay, so I think if anyone have any question but is too shy to ask, you can stay for a while. But I think we can stop today's seminar right now. So let's thank Dr. Revzen for giving this great talk. And also thanks everyone for coming. I'll see you in our next EEB seminar. Okay.
Thank you.